Again, thank you everyone for joining us today. We're excited to have you here at our webinar on the Allen cell collection. So please use the Zoom chat box to tell us where you're calling in from and what you're excited to learn about at today's webinar. If you have any questions for our presenters during today's talk, please use the Q&A button here in Zoom to ask those questions. If you're joining us on YouTube, there is only one chat box. There is not a chat and a separate Q&A. So just go ahead and put all of those chat and Q&A messages there. We will take questions from both locations, and those questions will be answered at the end of today's webinar. So again, those of you joining us in Zoom, please use the chat button to introduce yourselves, tell us where you're calling in from and what you're excited to learn about today. Make sure you're sending your messages to all panelists and attendees. And use the Q&A button, which looks like two conversation bubbles or speech bubbles, to ask questions for our speakers. We will answer those questions at the end of today's webinar. If you're joining us on YouTube, we will be taking questions there as well. So please go ahead and use the YouTube chat to ask those questions, and we'll transfer them over to Zoom. We'll give another couple moments for people to trickle in right at the top of the hour here. And today we are going to be speaking on the Allen cell collection, the open cell lines produced by the Allen Institute for Cell Science, which you can obtain and use in your lab, stem cell core facility, or other uses. So this is a Zoom webinar. We cannot see or hear you. If you would like to talk to other attendees, please use the chat box in Zoom or YouTube. And if you are here in Zoom, please use the Q&A to ask those questions. That'll make sure that those questions don't get lost in the shuffle. So we are now at 1 past the hour, so I'm going to hand it over to Caitlin Gerben, who is going to get us started.
Sorry. Hi, everyone. Thank you for joining us today. I'm Caitlin Gerben. I'm the Associate Scientific Project and Alliances Manager at the Allen Institute for Cell Science. And I'd like to just start us off today with a brief introduction to who the Allen Institute is and what we do before I hand it over to my colleague, Kim. So the Allen Institute is a biological sciences nonprofit, and we're located in Seattle, Washington. We have research focus areas in neuroscience, in cell biology, immunology, and we also support cutting edge research across the world through the Paul G. Allen Frontiers Group. All 4 of our institutes share a common goal in tackling complex, broad, and difficult problems in fundamental science. And we have 3 core principles that guide this approach. I'll return to these in a moment. But all of this work generates data, knowledge, and analysis tools, and we make all this stuff available to the public. So on the next slide, as I mentioned, our research is guided by these 3 core principles. The first is team science, and this means that we rely heavily on interdisciplinary teams, all working towards a common goal. We use constant collaboration and communication to do this. Big science is the next one, and that means that we take on large-scale projects and generate massive and useful data sets. And to date, the Allen Institute as a whole has generated over 14 petabytes of data. And fun fact, just to orient you, that's enough to fill 2.8 million DVDs. So big data sets. And lastly, our approach to open science, I think, is what really makes the Allen Institute unique. We share all of our resources with the public and the community, and this includes all QC data, analysis tools, and products, including our cell lines. So with that, I'd like to introduce my colleague, Dr. Kimberly Cordes-Metzler, and Kim is going to dive deeper into one of our open source resources at the Allen Institute for Cell Science. This is the Allen Cell Collection. So Kim, I'll hand it off to you.
All right, thank you, Caitlin, and I'm happy to be here today and glad to see a lot of people participating. I'm going to get started. I'm going to introduce our Allen cell collection, which is a collection of fluorescently tagged human-induced pluripotent stem cell lines to help investigate cell and nuclear organization during differentiation, disease, and regeneration. Oops, here we go. One sec. Okay, there we go. All right, well, so here at the Allen Institute, our research goals are to create a multidimensional map of stem cell states, such as mitosis, differentiation, and looking at perturbed states, with a focus on 3D live imaging. Our goal is to create this human stem cell state map. Now, there's been lots of research in the recent years on genes and proteins and how they influence disease or phenotype, shown on the right. But, there's really a gap in the knowledge of that intermediate step, the structures and organelles, meaning like how do those genes influence the composition, shape, and functioning of cells? And how do those variations really affect disease? So to answer these questions, we realize that we really need to build the tools and resources that could help us fill this gap with the outcomes that could better define cell types and to define new rules of morphogenesis through cell transitions or fate decisions that could allow us to predict normal or disease-specific behavior. Now, we chose the human iPS cell as our model system because they are human, diploid, they're easily grown in the lab, and they have the ability to become all different cell types in your body. So we've focused on understanding the pluripotent stem cell state, and we began efforts of looking at the cardiac state, which I'll describe a little bit today. Now, see here. Okay, so where do we start? One of the, what we're trying to do is kind of map the landmarks of the cell. And so we started to ask questions such as where are the major locations of the structures and what are the morphologies of the key structures and organelles of the cell? So I mentioned we took this approach of 3D live cell imaging using high res and at high scale. And we wanted to develop real integrated cell models that represent different states a cell could be in, like during cell division or differentiation to a defined cell type. And we asked, what is the normal variation and relationship between structures? And how does gene expression really correlate to organizational changes? Our goal is to build realistic integrated models that represent the cell more accurately than the textbook models that you see here on the upper right. So what can we learn from the landmarks of the cell? We can start to, ask the question, is it the ER that drives the mitochondria to organize for organization or vice versa? Or how do these cells transition between states? With image-based analysis, we can start to understand mechanism and make predictions using 3D cellular models. So So about 5 years ago, we began to create the foundation that would allow us to map the landmarks of the cell, which brings me to creating the Allen cell collection. So this collection is a suite of fluorescent protein tagged iPS cell lines, whereby we get input from the community experts for each structure. And this collection of gene-edited lines is created in the same background or parental line, which is the WTC11 HIPSC cell line that's derived from a healthy donor and generated by Bruce Conklin's lab at the Gladstone Institutes in San Francisco. Now, we chose this line because of its great pluripotency and effective differentiation into multiple cell types, including the cardiomyocyte lineage as shown here. And with this collection, we perform dynamic high-resolution 3D imaging to begin our mapping of the landmarks within the cell. So we make all these resources publicly available to the research community through our website, allencell.org, which includes our cell catalog, our methods and tutorials, and our discussion forum. So today, I'm going to walk you through this workflow on the top of how we gene edit our cells, the quality control process, a little bit of the cardiomyocyte differentiation, and then take you through a demo of our website so you can find where these resources are. We've created this extensive list of resources because we want to empower you guys to do your research and, and help you along the way. Okay. So, so why use our Allen cell collection? Well, there's multiple reasons you can think of, and I'm just going to list a couple here. So as we as an institute follow our mission to understand the organization of the cell, we really felt this collection could be useful for many other researchers. So we wanted to share it with the community. One reason is we put a lot of effort into standardizing our processes and spend time with quality control for each of our lines. Now, also these lines are very applicable for live cell and dynamic imaging. Shown here down below, you'll see images first on the far left. You'll see these are published works by different researchers that have been using our cell lines. And the bottom left is an example from an investigator, Chris Chen, where his lab has characterized the fundamental mechanisms of sarcomere formation in HIPSC-derived cardiomyocytes. And in the middle, you'll see a video from one of our collaborators, Bino Friedman at University of Washington, who uses our lines to understand kidney disease and regeneration. And then on the far right, you'll see a new study that just came out from a group from Austria, where they're creating these cardiac organoids called cardioids. And this is an example of a live imaging of that cardioid from over 24 hours. And what they found is that they can kind of recapitulate a cavity formation similar to the chambers of our heart. And so this allows, the study of cell biology, development, organoids models as shown here, and even disease modeling approaches. You can observe structural dynamics and integrate structural organization. These cell lines are also great for drug screening readouts. You can imagine that different structures can be, if they're perturbed by the cell, you can visualize that and use that as your result. And then finally, what complements these cell lines is that we've developed our own tools that will help with image analysis for 3D images downstream of imaging our cell lines. And we've also made these tools available to the public. And so in a way, you've got a complementary tool set with our cell collection. And also we have our protocols online, and we have scientists on standby via our forum.allencell.org, that if you have any questions, our scientists are happy to walk you through any type of steps or processes or issues that you're encountering if you have our cell lines. And we're just trying to reach out to the stem cell community and become integrated with them. So as I mentioned, that we have developed downstream tools to help with image analysis. We've created the Allen Cell Explorer Toolkit. This is kind of an emerging suite of tools and workflows that allow for visualization and interrogation and modeling different cell behaviors in 3D. And so I've listed these resources here, which are also available on our website. Now today, I'm just going to focus on the very top one, the Allen Cell Collection, but stay tuned for future webinars as I will discuss these other topics later on. But also, if you want to know now, you can go to our website to learn more. So coming back to our Allen Cell Collection, I'm going to first focus on our gene editing approach and our QC process. Okay, so to achieve the desired knock-in, we use the CRISPR-Cas9 system and the ribonucleotide protein, or RNP method, to transfect the fluorescent protein containing donor plasmid, which will introduce the tag endogenously at a specific loci. And it'll allow it to be a really good reporter for us to look at a specific structure or organelle within the cell. Now, for example, to enable the observation of microtubules, we targeted alpha tubulin with GFP. Now, next, we enrich the edited cells expressing the fluorescent tag by FACS, and we generate clonal lines that we genotype in QC to look whether it is a monoallelic or biallelic edit, or if any imprecise editing may have occurred. Then we spend a lot of time and effort to analyze and perform quality control. So shown here. And this would make sure like the tag doesn't perturb the cell and that the cell line behaves as expected. Like we look for differentiation into another lineage, and such as cardiomyocytes and so forth. Now, this process takes about 6 months, and here we go. Whoops, there we go. It takes about 6 months, and in this approach, because we perform a gentle editing approach using our RMP method, we've been successful for greater than 50 edits, which again are mainly monoallelic edits, but we have mainly single tags, but we do have a couple of dual edited lines and triple edited lines. Now, we deposit our donor plasmids that contain the GFP at Addgene. And so here, I'm just showing kind of the web page of Addgene. We have an Allen Institute page that describes or has the catalog of plasmids that we have deposited there. So let me just play this real quick. So if you kind of scroll down and see you'll see a list of our plasmids with the most popular plasmids listed first. TOM20 is one of our most popular. It's the mitochondrial line. You can get details. You can click on this link. You'll get details of our homology arms that we use, the linker GFP sequence, and other information. So for example, if you want to target a different cell type other than iPS cells, or if you want to add this tag to a patient-specific line, maybe a patient-specific line that has a mutation, this resource is available. And I'm going to describe in a little bit links to our protocols that could walk you through using this and putting it into your own cell type. Okay. There we go. Oops. Okay. So coming back to the gene editing workflow, I mentioned we spend a lot of time with the QC process. This can take up to 2 months and sometimes longer. And so this extensive QC process is really taken to validate our edited stem cell clones. So I'm going to walk you through our QC steps in some detail, but want to mention that I'll show you on our website where you can find this information for each of our cell lines. Okay. So after FACS sorting, we get as much as 90% enrichment of fluorescently positive cells, and we pick around 96 clones and ask, is this tagging precise? Now, we developed a primary screen that uses digital droplet PCR and junctional PCR to check all clones in parallel. And the readout is to make certain the fluorescent tag is in frame with the targeted exon and to make sure that there's no random or on-target insertion of the donor plasmid's backbone. Now, of course, we look for unintended mutations in either alleles, and we asked, is this tagged protein expressed and localized to the correct structure? And we do this by observing the morphology by imaging and through immunostaining and looking at level of protein expression. In addition, we do typical analysis of stemness characteristics and normal karyotyping on each cell line by asking questions, do these cells behave normally, and are these cells pluripotent and stable? And so once a clone has passed all these QC metrics, we scale up in-house and send these vials to Coriell for distribution. Okay. So here I'm just linking to the Coriell's website. Coriell is a biobank containing multiple different cell lines from different people. And shown here is our Allen cell collection. Now you can read more about these lines here. And they also offer a core facility agreement if you were to go here. And you can, as I'm going to play this, so if I just scroll down on their website, you can find one of our cell lines. And if I click on, say, lemon B1, you can find out more information about this cell line. And we also ship a certificate of analysis with every cell line that is ordered. And we contain the certificate of analyses on our website so that people can see all the results from our QC process. Okay. Go back here. All right. So here's a full list of our available cell lines. We've created 51 so far, and it's rather than squint at this list, which I know is small, feel free to either write this link down, allenscell.org/cell-catalog, or you can take out your phone, open your camera, and a link will pop up in that camera window to take you directly to the cell catalog. So I'm going to show you the cell catalog in a few, and we'll go over it live and just kind of walk you through where you can find this information. Because again, we got the feedback from the community that from different researchers that they wanted to know a lot about what we do, what our QC process is, so they can think about that in their own research needs. And so we've provided that on our website for your use. And we'd love to hear feedback about any of that. Okay. So now I want to just kind of show you highlight some examples of our lines that we've imaged using a spinning disk microscope. For example, this is an alpha tubulin cell line that highlights microtubules in the cells in the pluripotent state. So in this image on the left, you can see we're capturing colony dynamics, and you can see some cells undergoing mitosis. Now, if you zoom in on the right, you can see this one cell undergoing mitosis and dividing, which is, really great. These lines provide really great imaging. So if I'll show you another example, this is using our Layman B1 line, which marks the nuclear envelope. And if I click on here, we can observe so in this video, we can observe the breakdown of the nuclear envelope and the reformation during mitosis, which is, you can see those cells breaking apart here. Okay. Let's see, click, okay. Now, in fact, we've observed how 15 of our structures, so I'm going to kind of play these videos each here. We've observed how 15 of our structures from our imaging kind of change during mitosis, and found that some organelles reorganize, others disappear, some others stay intact, like you see in the middle and on the far right. And what we found is we can integrate all these 15 different structures from our different cell lines, and this can be observed in our integrated mitotic cell story, which I've just kind of highlighted an image here on the bottom right. That is our integrated mitotic stem cell. So you can find out more information about that. I'm not going to talk about that in detail, but this is just a great way to observe cellular behavior using live cell imaging. Okay. And then we love to keep track of how the community is using our cell lines. We feature cell stories on our website to kind of showcase new discoveries. And also, I want to highlight on the left, we are connecting with many universities and institutes that have stem cell core facilities to kind of offer our cell lines to more investigators. So if your university core facility isn't listed here, feel free to contact us or me via our website, and we can talk further. But it's for some really exciting work going on, and we just love hearing about it because it's fun, and we're just making new discoveries in medicine and research, and it's neat to hear. So I want to then kind of state that as part of our 51 cell lines that we've created, we've actually dedicated effort to create nuclear cell lines or nuclear specific lines to better understand the nuclear landscape, and to begin to build integrated models that combine structural imaging with genomic organization, as you see here on the left. This is a collaboration we're doing with the 40 Nucleum, which is an NIH Common Fund program. So here on the right is a list of 17 nuclear structures and substructures that we have created so far. So I just want to show you some examples. So here are 8 examples of these nuclear lines. So you can see the kind of diversity of nuclear substructure localization we are capturing to help understand nuclear organization and reorganization during transitional state changes or differentiation. Now, we even created a dual edited nucleolar line combined with a DCAS9 CRAB that's expressed in the safe harbor locus. This is a CRISPR-I tool line that we used in our own work to identify novel genes involved in nucleolar morphology. So here on the far left, we're combining kind of a pooled screening method with a FACS-based readout that's based on capturing the width of the fluorescent signal, whereby cells with normal nucleolar shape and size will be enriched in these middle bins 2 and 3, while cells that are condensed or diffuse will show up in bins 1 and 4. Now this will kind of allow us to observe nucleolar changes and efficiently and simultaneously screen and analyze the 20,000 genes in a primary CRISPR-I screen. So you can see that here, we kind of plotted that out, and you can see those that are captured in red and blue that are either diffuse or condensed. And then we're now performing a kind of hit validation screen with an imaging assay to validate our hits. So this is just one use case for this line. We also have the CRISPR I line available too, but I just want to come back now to the creating the Allen cell collection. So I don't know, quick. I lost my cursor. There we go. Okay. So now I've told you about the gene editing process and the quality control that goes into making our cell lines. And I gave you a preview of the lines we've created so far. But I also want to point to an area that we have focused on with cardiomyocyte differentiation and producing cardiomyocyte-specific lines. Okay. So we use a small molecule protocol that involves the inhibition of Wnt activity to differentiate our iPS cells into cardiomyocytes. And we're interested in understanding the state transitions during this process and how the cardiomyocyte becomes organized also through this process. And we first designed a strategy to create gene edits with sarcomeric genes. Since sarcomeric genes are transcriptionally silent in the pluripotent state, we developed a multi-step editing process to tag genes that are not expressed until differentiation. So we designed a donor plasmid that contains a GFP and an M-cherry cassette that's driven by a constitutive promoter. Then by enriching for M-cherry positive cells, we next excised the cassette with Cas9. And then by strategic design and guided microhomology mediated end-joining, with the GFP tag intact, we were able to enrich a GFP gene-edited population that has our cardiomyocyte gene intact. So this process can apply for tagging any type of transcriptionally silent gene that's in the pluripotent state. So any type of gene you want to differentiate to. And here on the right is a table of the different sarcomeric protein or sarcomeric genes that we've tagged. And then, sorry, here we go. Keep losing my cursor, I apologize. All right, there we go. And so these cardio-specific lines provide opportunities to follow sarcomere formation and maturation and to study disease mutations. Okay, now with tagging numerous parts of the sarcomere, we knew we could begin to stage and classify different types of organization. Now, starting with our alpha-actinin 2 line, which marks the Z disc of the sarcomere, highlighted here on the green, this line, we could stage and automatically classify our cardiomyocytes based on a machine learning approach that looks at alignment of sarcomeres and how organized the sarcomeres appear, yellow being the most organized and purple being the least. And we applied this classifier with analysis of gene expression, of single cell gene expression, of RNA, of fish gene expression to over 30,000 cells in a recent publication called Cell States Beyond Transcriptomics. And this study highlights a framework to integrate gene expression with our imaging data. So these analysis tools are described in the paper and are also available on our website and in the Cell Systems manuscript. Okay, now moving on to, so coming back here, I want to now focus on our publicly available resources. I want to first introduce our cell catalog. So here we go. So I'm going to move over to our website. And I'm going to give you a demo of our of how to find our cell catalog. So if you go to allencell.org, you can see that if you click on the Allen cell collection, you'll see our mega menu, which you can select different things. We're going to first start with our cell catalog. If I click on that, you can see there are always updates, the latest updates of what new lines are available. And then we can scroll down and you can see the different list of the proteins that are tagged. You can find information of which, where the tag is located, so on the C or in terminus. You can also filter by what structure you're interested in. So let's say that we're interested in a structure. I can just pick arbitrarily like microtubules because you've seen the images of that. And you can see there's two, it narrows down to two. One, the difference is that one contains a GFP tag and one contains a red fluorescent protein tag or RFP tag. And so we have tagged some of our lines with RFP, but most of them contain a GFP tag. So if I click on this link, it's going to bring up a page. It'll have a Z stack, sorry, of that cell line, and then I'll have information on the left that contains everything from a data summary of our QC analysis results. It'll have a link to the Coriell website to get the cell line, or a link to the Adgene website to order the plasmid or get more information about that plasmid. We also have a link to our certification of analysis certificate that is also shipped with each cell line from Coriell. Now you can scroll down and you can see more information is here. So not only is that information contained in a PDF, if you want to print it out, take it to your lab bench and then take a look, or if you're on the computer, you can take a look and add it in more detail. So here we have our editing design strategy of where we tagged the GFP. We have information like the CRISPR RNA sequence, the linker. And if I click on now genomic characterization, you can see here is a schematic of the amplified junctions for our junction PCR and the results below. And just kind of walking you through here, cellular validation, we can look at, protein expression or localization by immunostaining, and then going to looking at stemness markers, we can see the state of pluripotency for those cell lines and including the differentiation potential for the different germ layers and cardiomyocyte differentiation. So we provide all our protocols also online, and we update them regularly if we make any type of changes to them that might optimize the technique. And those are continuously, you can find those here through our protocols link. There's another way to get there, and I'll show you in just a moment. So I'm just going to take you back to the slides. There we go. So I'm going to come back here, and now I'm just going to talk about the methods and tutorials. So I showed you where the cell catalog is on our website. Now the methods and tutorials are also available. Now I alluded to the protocols or methods from here. If I come up here, I already have this saved in my browser, but you could get to it from here, going to lab methods and clicking on instructional videos, standard operating procedures or written methods or illustrated overviews. So looking at the instructional videos and tutorials. So what we've done is we've had our scientists from the different teams from gene editing and assay dev and our differentiation teams, capture some of the methods in a video format so that people can really follow the process. Because sometimes it's tricky. There's little tricks that you learn in the stem cell field about like an angle to hold your cells, or things like thawing human iPS cells, culturing and maintaining them. And we've got the RNP transfection, and then we have our Jove video journal article that highlights the paper where we described our tagging approach using CRISPR-Cas9. We have a cardiomyocyte differentiation video tutorial and techniques with working with nitrogel and cryopreservation. I'm just going through and naming these. And then we also have our newly released kind of automated tissue culture platform, which if you're interested in, this kind of showcases an automated tissue culture platform to help seed and passage, feed, and maintain your cells. And so you can look for more information on here. Now, if I scroll back up and I go back to the Allen Cell Collection, you see also there's a link for our methods, and there's illustrated overviews. So I'm going to head back. So you're welcome to look at those for more information if you're interested. And then I'm just going to, Now our next part I want to get to before the end is our discussion forum. So I alluded to this during the talk, and you can visit our forum on allencell.org. And this forum is free to use, but we do ask that people, if they want to ask questions, to sign up for it, and which you can find here. You just sign up, you click the sign up, and it'll ask you for your email and all that, but all that information is private, stays private. But the reason why we do that is so that we don't get any like bot spam, and asking questions that aren't appropriate for that discussion. So it's just a way to keep that a safe discussion. But you don't have to log in or sign up if you just want to browse the questions and see what people have written, what topics are popular. You can hit the latest tab. That'll give you the latest topics that have been discussed, or you can pick something more specific to your interest, whether it's gene editing, culture techniques, some of our imaging software and code, which I alluded to, and I'll talk about later in future webinars. But you can just click on one of these. I'll click on gene editing, and you can see some of the top topics that have been discussed. And so that's our forum, and I think it's very useful because what we've done with this forum is we've kind of automated any type of question that gets asked goes directly to one of our like a group of scientists or engineers to answer that question. And so they typically answer within 24 hours, pretty quick response. Now, coming back to here, I want to just kind of end with a summary of, we've created the cell lines, to answer our own scientific questions. We think we find a lot of value in this resource, and we hope you do too. And so we have been taking the step of going from, large scale 3D imaging, and next we want to go towards single cell information. So here is, let me just play this here. So here's just some Z-stacks that go through, some of the cell lines I discussed today. And, we take images. Now we've imaged over 25 structures and counting. And we're starting to think about integrated ways of taking all those structures together. And I'll talk about some of that in the future webinars. But really what I want to kind of highlight is that, so we go from this microscopy, we take the images, and then we go through some image processing steps. So just like people that are studying single cell genomics, now we're thinking about single cell imaging. And we go through an image processing step that then can segment the cell, the nucleus, and the individual structure of the cell. And so I just want to mention that the next webinar will not be until September, but it's going to be on the Allen cell and structure segmenter. So it's going to lead into how you can take your imaging data that you get and actually start to analyze your images in a single cell way. And just think about the potential of this information along with your single cell genomic information and other single cell information that is being widespread, how powerful this can be. As I always hear, a picture is worth 1,000 words. And so here are these cell lines do produce, beautiful images. So with that, I want to end and thank the cell science team. Now, specifically, I do want to give a shout out to the gene editing team, which consists of our differentiation specialists and our gene editors, and as well as our assay development team for, they've really been the key people that have developed the lines. And then the rest of the team, thinks of ways to use those lines to get, to analyze and to answer our scientific questions. And we all believe in open science, team science, and big science. And so it's a fantastic group to work with. And then finally, I'd like to thank the Allen Institute founder, Paul Allen, for his vision, encouragement, and support. And so with that, I'm going to end and take any questions. So thank you for your attention.
Thank you, Kim, for a great talk. I'll just remind everyone that we've have the Q&A function open, so please submit your questions through that if you're watching via Zoom. You can also message in the chat if needed to the panelists. And if you're watching on YouTube, go ahead and put your question in there. We're pulling questions from that as well. So Kim, we've got a number of good questions coming in. So I'm going to start with one from the chat box. So what is a type of laser microscope used in imaging our lines?
Yeah, so we use spinning disc confocal microscopes in imaging our lines. In particular, we've used Zeiss and a 3i imaging system.
Awesome.
Yeah, thanks.
And I'll just add to that if there is a specific line or a specific edited line that you're interested in for your lab, and you want to make sure that the imaging capabilities that you have are efficient for that, go ahead and ask those questions in our forum. We have the scientists that can have been imaging those lines and can give you a little bit more information about what specific specifications you might need.
Oh yeah, and I'd like to add to that too. We have had questions before that, people may have gotten our line and it might be dim in their scope, in their system. So yeah, reach out, because we love to help you with that and make sure that you can actually visualize a line that you've gotten through our collection. Yeah.
Great. Another question from the Q&A. Do you check for unspecific Cas9 cutting outside of the intended targets as part of the QC process?
Yes, we do. We have not gotten, because we use that kind of gentle RNP method, we've had no off-target effects that we have tested for that rigorously. I can ask my gene editors for how much they've done that, but I'm pretty certain that they've taken a pretty good doing a lot of research on that, and we haven't seen that.
Mm-hmm. Yeah, and I can add just a little bit more context to that too, that we do the PCR amplification and Sanger sequencing. And so that was done extensively to characterize for the first 10 lines in the Allen cell collection. And because we didn't find any off-target editing with that method, we don't routinely do that as part of the quality control workflow, but we're confident that the editing is precise and that there's no off-target event or off-target edits in that. There was just a question that came through about, I think actually could be great for thinking about the next forum. So she wanted to know, what are some of the next developments for the segmenter?
Oh, that's great. So I didn't mention the segmenter in detail, but we have a kind of a classic segmentation workflow and a machine learning workflow that take advantage of our cell collection. Because we've got so many structures represented, we've created structure specific workflows for each of our 29. So 29 structure workflows is what we've developed. And so we can really, so one of the developments I'll just allude to is that we are making it more user friendly, because right now, it does require some programming skills in like Python and Jupyter notebooks. But now we're going to make it even easier for scientists to use. So on June 30th, we'll have an announcement that we'll have an announcement of how we've kind of increased or better made the user interface more accessible. And I'll talk about that in September in more detail. Yeah.
Great. Thanks again, everyone, for adding the questions. We're going to keep getting through some of these. So again, feel free to drop your questions in the chat box for us. So there's a question here. What kind of approach would you use to integrate single cell structure information with other single cell omics type analysis?
That's a great question. So we thought a lot about this, and we did just, that paper I referred to on the cardiomyocyte, like classifying and then trying to integrate structure or genomic information with that structural information. So one thing is that you have to segment the cells to get that single cell image, right? And then you have to kind of know what information or what type of assay you're going to do for the genomics. So we used RNA fish, and we tried to look at genes that were expressed that might affect structural differentiation or structural organization. And we did a process to kind of verify which genes are kind of the highest expressed in different time points. And so we then kind of try to correlate that information using kind of a linear regression model and some statistical approaches to incorporate that information and see what so there's, and I was going to say, and the other part we're doing that is with the 40 nucleome project, we are also thinking about how to integrate single cell information with single cell genomics, for example, and other information. And although we have come up with some strategies and ways to tackle that, it's still an exciting field to get to kind of jump in, because I think a lot of people are starting to think about that. Caitlin, did you want to add anything to that?
Yeah, no, that's great. I think, like you said, I mean, we're an imaging-based institute, so our approach was imaging-based. So that's why I think one of the reasons why we naturally gravitated towards the RNA fish, so that we can integrate that structural information. Another thing, just along with that paper and the data set, so all of that is available. You can find it through the links I think were dropped in the chat earlier in the talk. And there is a ton of really cool science going on there. And, I would say, found some amazing things, but also kind of just scratched the surface of it. And our hope is that others in the community can go and use these toolkits, analysis, Jupyter notebooks, and data sets that we have available to actually do some more of that information. So I think potentially there's a lot of really cool stuff to follow up on in there. And we hope that the research community is excited about that data as well. Great, so let's get to a few more questions. Do the tags affect the differentiation success?
No, we have not had an issue with our tags with the differentiation. In fact, if they did fail, it would not pass our quality control that we put onto the cells.
Great. How about another one here? Can I use your tag in my lab's own cell lines?
Yes, definitely you can use. So I refer to the Adgene's website that you can find where we have deposited our donor plasmids. And so you can definitely look at that and see if that would work for your cell type. And we're happy to help with that as well, because we love to learn about what people are doing with our plasmids as well as the cell lines, because it's, and like I said, we have a team of people dedicated to doing this. So we want you guys to be successful as well. So just reach out if you guys have questions.
Awesome. There's another question in here about the cell lines and the edit choices. So can different research groups request specific cell lines to be made, or like what is our process essentially for choosing which edits we do?
Yeah, that's a great question. We get that question a lot. So we initially started out, asking key experts in the community, what would be the best represented gene to target to capture that organelle or structure that we're interested in. And now we kind of do for any future cell lines, we still do the same thing. We look for, key stakeholders that, are, and kind of, we love to get, if we hear the ideas enough, we may, if someone says the same gene over and over and we hear that 100 times, that does definitely get on our radar, but we don't take personal requests to create specific cell lines.
Yeah, and I mean, the goal is we're trying to create tools and resources for the broader scientific community. And so we're trying to make sure that we're covering that. So yeah, no specific requests. Okay, let's see, get to a few more questions here. Sorry, one second, just scrolling through. Where do you karyotype the HIPSC lines? And if so, where do you get that work done?
Oh yeah, we do karyotype our lines. I think it's called, is it DCI? Is that?
Yeah, Diagnostic Cytogenetics. They're a local Seattle company.
Yes. And we have all our karyotyping information, as part of released with our QC data, and you can see the images on our website, the karyotype analysis on our website.
Great. There's a question from the Q&A here. Do you take images over different inertial environments, such as in centrifuges? And I guess maybe you could add to like what type of platforms are we using for most of the imaging?
Yeah, so we have not done that. I'll start with that. But we do, so what we do is we take, we kind of grow our cells in 6-well plates, and then we transfer them to 96-well glass bottom plates. So we do image our cells on glass for better better optics. And so we have a protocol of how we image our cells on our website, so that you can see, you know, like how, if you wanted to do that and image them in that way, you could kind of replicate our strategy. And it works well for our, you know, we've optimized the Matrigel and the, you know, so that's I think a good resource to get there, but we haven't tried, you know, imaging in centrifuges or any inertial environment, and not that I'm aware of, but I'm pretty sure we haven't tried that. Yeah.
Yep.
But thank you for that. That's interesting. It's an interesting.
Yeah, I think there's a lot of interesting different ways to do the imaging, and like, you know, we've been optimizing, like you said, for kind of a 2D imaging platforms that we can get high resolution of the cells and the structures on glass. But of course, there's so many different environmental factors and stuff that we could look at for different ways to, you know, study cell behavior with the structures. There's a question here about the gene editing process, and actually I'll combine kind of two together. So there's a question asking, where in the gene editing process do you see most clones fail? And also if you could add a little bit more detail about like, how do we confirm precise editing through that process, and where can people find more of that information?
Yeah. So those are both great questions. So when we get that frequently. So first, where does it fail? It usually fails right at the kind of first step, which is when we do so we'll identify it usually at the first step. When we do our junctional PCR and ddPCR, we can see at that step if something's failed. That's typically where that genetic analysis. But then the second part of the question, remind me, Caitlin.
Just how do we confirm precise editing?
Yeah, so we do confirm it by using that method. So one, by using the junctional PCR and ddPCR, we can confirm, you know, whether you know, our copy number of the GFP that's there. We'll do sequencing to look for any mutations on either allele, and we make sure that the backbone hasn't been integrated and that the GFP has been inserted in frame with the exon. So we do, you know, those type of assays to really look, and that's where that's where we'll see most of our clones fail, is at that first step. And then after that, you know, we've had pretty good success, but, you know, it's, I think like about 1%, you know, at that point that move on. So.
Great. We'll just do a few last questions here. So question from Chris, has QC been done to see whether tagging changes the expression or localization of the proteins?
Sorry, has what been done?
Has QC been done?
Oh, yes.
Localization and expression levels of the proteins.
Yes. So we do a Western blot analysis. We do immunostaining. So we take an antibody that's a really well-established antibody for, say, a specific protein that addresses, you know, the specific structure that we want to look at, and we'll compare our live image of our line compared to that immunostain. We'll do live and fixed cell imaging, and then we also do a Western blot to look at the expression of the endogenous protein with the you know, with the GFP as well, to see what the differences are. So we have all of that, you know, we have all of that represented. So, you know, all kind of transparent on our website, so that we make sure people are informed of what they're getting in that cell line. Yeah.
Thanks.
So
yeah, that's a good question. Okay, another question about differentiation. So you showed the cardiomyocyte differentiation, but what lineages have been created with these cell lines, and what lineages have we differentiated in-house?
Yes, that's a good question. So we've really focused on the mesodermal lineage and differentiating in the cardiomyocytes, obviously, as I showed, but we do a trilineage kind of potential assay to make sure that it can differentiate into endoderm and ectoderm. We haven't taken it like farther out to like long-term like neural cell types, but we have followed up with researchers that have had success in doing that with our lines. So we do keep track, as I mentioned, of what other people are doing. It's hard to keep track of what other people are doing, so we love to hear from you if you are using them. But of those people that we can, that come out to us or ask questions, we can see that they've had success in it. And we do work with people that do have trouble. If they have trouble, we try to help them solve that, just by giving advice and things like that and follow through. So, yeah.
Yep. And I'll just add to another plug for the forum there, that would be a like perfect kind of question or thread to put in there if you're interested in a specific differentiation lineage, that you can try to get help from different people in the community who are using that and maybe help with protocols and stuff.
Yeah, and that reminds me that with the forum, I do want to mention that it's not just for people using our cell lines or our tools, it's for the stem cell community. We've seen other people like answer each other's questions and respond. So it's kind of a network of people doing stem cell research or gene editing and coming together to kind of ask questions. So yeah, you can utilize that for, other stem cell specific or gene editing specific questions.
Great. So last question, not as much of a question, but I just want to circle back a little bit to the imaging, since we show obviously so many beautiful images and stuff of the cells on our website and also in the presentation here. So I wonder, could you just give a little bit more detail about the imaging that we do, and also maybe a plug for future imaging webinars and analysis tools that we want to share with the community?
Yeah, definitely. So, our imaging platform, like I had mentioned, we use spinning disc confocal microscopes to do our imaging. We take 2 different magnifications of our images. We do live imaging. We also we are now also capturing some videos to capture dynamics. And we, as I mentioned, on an image on glass, 96 well glass bottom plates, and we take several fields of view of our plates so that we get different representations of the cells within so in the stem cells, for example, within different colonies that are grown on the plate. And we capture all the associated metadata that is with that. So for example, like, what cell line, what day, I'm trying to think, like, we have like how dense the colonies are. So we try to capture all that information and share that, because it might be useful for people downstream in the way they're trying to think about their analysis approach or whatever. But we put all those images that we've taken now, and I'll mention this down the road, but in future webinars, but we've taken now like over 18,000 fields of views of our images to capture the variation that we get between in the stem cells with each line. And so we have thousands of images of each line, and that's also on our website in the Cell Feature Explorer and available for download or just browsing and looking, and you can kind of see the types of features that we calculate on those cells, like volume or structural volume and so forth.
Awesome. Okay, well, thank you, Kim, for answering all those questions. And thank you again for everyone who followed along. Thank you for submitting your questions. I'd like to just conclude with saying that we really appreciate input from the community and engagement. And so please follow up with us on the forum if you have any follow-up questions. And also, if you're interested in learning more about some of our other tools and imaging, please register for our forum our webinar that's coming up in the fall. We'll be going over more of the segmenter toolkits and also have future webinars that are going over imaging and things in more detail. So with that, yeah, thanks. Thank you, Kim. Thanks everyone for joining us, and we hope to see you in the next webinar.